So this is the classic experiment that basically proved that DNA replicates by semi-conservative replication, and it's how we know that this is the sort of the way it does it, and it's one of the first ones. So they grew E. coli in a medium which was high, which had the heavy isotope of nitrogen 15. And they grew them there for, many generations until this nitrogen was incorporated into the DNA. And so you would think that both strands of DNA would be the heavy type. And then what they did is they moved them to a different medium containing the lighter isotope, the normal isotope of nitrogen, N14. So they're E. coli, I said that already, and the E. coli sort of replication time is around 50 minutes. So every 50 minutes, one cell splits into two. So E. coli don't have nuclei, they have a little bacterial sort of chromosome. That's what that is, not a nucleus though. And then what they did is they took this mixture and they centrifuged it. So in a centrifuge you have a tube like this, you put in all your stuff and then you insert that into here. So if this is the centrifuge, what happens is it spins around like ridiculously fast. It basically spins everything really fast, and then whatever's in the tube separates by density. So this is the heaviest and this is the lightest, so the least dense. So we get the E. coli growing on here. We expect all their DNA to be like that. And then when we move it to the nitrogen, we'd expect it, because of semi-conservative replication, that we have this splitting into one of those and one of those. And then we have our light isotope forming the new DNA. And then the next generation, we would expect to have one of these for each of those, plus one of these for each of these ones, and then we have our new isotope, so it'd be like this. So in the second batch we'd expect to have two sort of new DNA strands which are completely the light isotope, one which is, and two which are half and half. In the first one we'd expect it to be half and half, and in the in the first generation, then the initial one, like at 0, so the parents would expect it to be all heavy. And not surprisingly, that's exactly what they found. So when they transferred it from the heavy isotope to the light isotope, and then they took it out straight away. So the parents, so the E. coli, they took a sample, they centrifuged it, and this is the tube that the centrifuge was in, and they had a band. And so we're going to say, okay, well if that's the parent strand, then that's this. That's our two heavy strands. Then 50 minutes later, after 50 minutes, they took out some more cells, centrifuged it, and looked and see what they saw with the DNA. And then the first generation, what we said was that there would be one heavy band and one light strand. So the DNA overall formed a band around there. And you can see it must, because it's higher up, it must be lighter than this one. Then the second generation, what did we say? Okay, so we said there'd be one of these again, but there'd be another one too light. And that's exactly what they found. So there was one where both strands were light, so you have a band up here, so it's less dense, and they had a band where it was the one heavy, one light again, so in line with that. So we'd expect after 20 generations, we've had loads and loads of these sort of being split apart, so we've got lots of our light strand as the template and then lots of light strands being added on, but whereas we're still going to have only the one or two initial sort of dark strands, so we'd expect a really massive band at the top and a teeny little band there, because we've got far less of the mixed one with the heavy isotope and the light isotope than we do with just the light isotope. And that was sort of the conclusive evidence to say, yeah, you know, semi-conservative replication, we have our two parent strands and then we get our new strands forming there. Like that. And this was sort of the evidence that showed that, so it's really cool.